Okay, we just finished talking about Western blot, ELISA, and immunofluorescence microscopy methods, right? And besides those, we'll talk about another kinds of method that will allow you to detect very specific protein-to-protein interaction happening inside of cell or outside of cell. Okay, and we'll talk about 3 different methods, as well as the analysis method that will allow you to detect between DNA and protein. Okay, so we'll talk about a couple of those methods. Okay, so then why is it important to understand what proteins are binding with another protein of different kinds? Because if you think about what's happening inside of a cell, there are so many different interactions happening inside of a cell. Okay, maybe outside signal molecules coming in and binding with the receptor protein, and then once that receptor is activated, and then maybe the receptor binding proteins like kinases may be activated. There may be cascaded of protein-to-protein interaction that may be happening inside of a cell. You probably learned about cell-to-cell communication or signal response process. So a lot of these protein-to-protein interaction is happening inside of a cell. That's part of your cellular processes. That's very important. So if you look at this diagram, it shows you some examples of protein-to-protein interaction that's happening a lot. So through all these interactions, what you receive from outside of a cell can be responded through maybe gene expression of different kinds. So there are many important protein-to-protein interaction that's happening inside of a cell. Okay, then how do you recognize all these interaction? That is done through these kind of analysis assay. Okay, so protein-to-protein interaction. What is it? Why is it important? Like I said, it talks about the interaction between the protein molecules because that is so important. It's part of the biochemistry and part of the signal transduction and part of the cellular network. Okay, so the interactions between proteins are very important for biological function. Okay, so outside signal that's recognized through signal and receptor binding, and then the receptor can turn on some other cellular responses through all these interactions. So proteins may interact very long time, or their interaction may be very transient. Okay, so all these things happen all the time, and some of those proteins are enzymes like kinases. Okay, so understanding protein-protein interaction is very important, like enzyme to substrate interaction, protein to ligand interaction, transcription factor to complex other protein molecules, how they interact, they're all important. So then what techniques are out there that will allow you to detect these kind of interactions? So among those, we'll talk about number 1, two-hybrid screening method. Name is kind of complicated, right? Two-hybrid, what does that mean? And what it means is basically you will create 2 different hybrid protein. In other words, 2 different fusion proteins. So 2 fusion proteins are created, and those 2 fusion proteins, when they interact, actually they will reconstitute one very important functional protein called GAL4. GAL4 is a yeast transcriptional factor. Okay, its main job is to activate the transcription of a gene that it is controlling. Okay, so GAL4 has a DNA binding domain and a transcriptional activation domain. Okay, so that protein will be split into 2 domains. Okay, so if I show you, so yeah, it's a GAL4 protein here, and naturally GAL4 has DNA binding domain as well as activation domain together. Okay, but you will split this single protein into 2 pieces. Okay, actually you do that through DNA engineering, of course, genetically. And then they are fused with other protein, maybe target protein and bait protein. Okay, they are fused to each part of your GAL4, DNA binding domain and activation domain. So the theory is if these 2 proteins interact together inside of a cell, okay, they will come together and bind. When they bind, okay, you will get your GAL4 kind of reconstituted back to its original condition. What that means is GAL4 DNA binding domain and GAL4 activation domain will come together. When they come together this much close, then they will function as normal. meaning they will activate the transcription of the gene that they are controlling. So if there is a gene that will be transcribed and translated, then that gene will give you some kind of signal. So this gene may be a GFP, or this gene may be some kind of a beta galactose enzyme, and that gene expression level can be easily detected. Okay, so it's kind of sounds complicated, but you do all this to understand what? The interaction between these two proteins. If the interaction happens, then yes, your GAL4 will be reconstituted. Then GAL4 will activate the transcription of this gene, and that gene transcription translation will give you a signal. That's what this assay will do. Two hybrid screening method. Okay. So again, same thing. So DNA binding domain of GAL4, activation domain of the GAL4. And then there's a bait protein, and there may be a possible candidate, whatever that binds with the bait protein. Okay, so you're interested in identifying, is this bait protein binding with any other particular protein, any special protein inside of a cell? That's your question, your answer. Okay, then how do you get the question? If you make a fusion protein between the DNA binding domain of GAL4 and your bait protein, and then any protein that you may think will bind with this bait protein, that will be fused with activation domain of the GAL4. Okay, and then you can detect interaction between this bait protein and this target protein inside of a cell, because if the interaction happens, then your GAL4 will be reconstituted, and you will see the transcription and translation of this reporter gene. You can detect that inside of a cell. So that's how it works. So normally this screening method is using yeast cells. So that's why it's called BIOT212 hybrid screening method, because you will create this reconstitution of GAL4 instead of living yeast cells. Okay, so DNA binding domain and activation domain of GAL4, each part is fused with the gene of your interest, that bait protein coding gene, and then whatever the target protein that may interact with your bait protein will be cloned on the activation domain of GAL4 coding gene. Okay, and then so that the interaction will be detected inside of a cell. So if I look at how it's done, so it's this way. So you have maybe three different plasmid in this case. So first plasmid, you have a maybe bait vector, meaning there may be a DNA binding domain, okay, and then coding gene. Maybe it could be GAL4 or LAX-A, whichever doesn't matter. And then there may be a gene of your interest, maybe the bait protein coding gene together. It's a fusion protein expressing gene right here. And another one is a prey vector, meaning there's maybe a activation domain of maybe GAL4, and then another possible candidate that may interact with your bait protein. That gene can be fused together here, so secondary plasmid DNA. And then the third plasmid DNA will have the reporter gene, and then the promoter sequence ahead of it. So if all these three plasmids are given into the yeast cells, and then see if that signal, in this case GFP gene expression, will take place or not. This gene will be expressed only if this reconstitution happens. Although this is LAX-A reconstituted with B42, but this can be GAL4 DNA binding domain reconstituted with its activation domain. But it's the same principle. Okay, if that happens, this gene will be transcribed and translated. You'll detect the signal, in this case the green fluorescence. Okay, so that's the first method. So this method, BIOT212 hybrid screening method, will allow you to detect the protein-to-protein interaction happening inside of a cell. So it's an in vivo protein interaction detection mechanism, detection assay. Okay, that's what it is. And number two method is called GST fusion protein pull-down technique. Okay, GST, we talked about it. It's a glutathione S transferase enzyme, right? You create a fusion of GST with your target protein, and then pull down. Pull down is what? It's a purification over a column. Now that's what GST fusion protein pull down is. Okay? How does that work? So GST coding gene is fused with a gene of your interest, which will code for your protein. Okay? Once that GST fusion protein is made, and then that protein, fusion protein, is probably pulled down through a column that has glutathione. So there is that GST and your protein of interest fusion together, and then you have the column that has what, glutathione in them. Okay, then glutathione will bind with the GST, and together your fusion protein is binding on the column, right? And then when you add entire cellular lysate, entire cellular proteins, if there's any protein that is binding with the target protein specifically, that interaction will be maintained on your column. Okay, so you can purify out this protein Y through that column. That's how this method works. Okay, that's why it's called pull down. You can pull down the target protein using this interaction. Okay, so you can fish out, you can purify out this protein by using a GST, the glutathione chromatography. Same thing again. There's a column that has glutathione attached to it. Okay, and then your bait protein, in this case maybe GST. Okay, so now you create that GST fusion protein, and now that any protein that binds with the GST fusion will be pulled down, purified over the column. That's how it works. GST fusion pull down assay. You can purify the protein that binds with the bait protein. The third method is called co-immunoprecipitation method. Co-immunoprecipitation. Again, immuno means antibody. Antibody precipitation method. Co, what does that mean? It's a together your protein is precipitated. So if you have a column that has antibody, and if that antibody specifically binds your target protein, yes, it's an antibody affinity column, right? Not only that, if you add the entire cellular lysate, entire cellular proteins, not only that protein binds with the antibody is fished out, also if there's another protein that binds with your protein or your target, if they interact together, you can purify out two proteins at the same time. That's why it's called co-precipitation. Two proteins are purified together. That's how it works. So again, it's in vitro purification, just like the GST pull down method. Okay, you're just using antibody to pull down, purify two different proteins at the same time. Same thing showing up here, right? Antibody column, all the cellular proteins are added. Then not only the antibody binding antigen protein, its target protein is also purified together. That's why it's called immunoco-precipitation. Again, in vitro assay. And the third method is called FRAT. Okay, we'll talk about what that is. First of all, you can see a GFP. It's a green fluorescent protein. It's a unique protein that gives you a fluorescent, green fluorescence. And normally that happens among those jellyfishes living the ocean bottom, right? But you can clone that gene that makes the GFP, and that gene can be moved to different organisms like a fly or fish or plants. You can make those insects or plants glow in the dark with the green fluorescence if you move the GFP coding gene into different location. So the GFP coding gene is cloned here in this plasmid DNA, right? And then if you can maybe clone your gene of interest that makes your protein, you can make a fusion protein actually easily. So you can create a GFP fusion. That's what this shows. So if you make a GFP fusion or any other fluorescent protein fusion, yes, using those fluorescent tagged fusion protein, you can measure the interaction of those two fusion proteins inside of a cell. We call that FRAT technology. FRAT. FRAT stands for fluorescence, resonance, energy transfer. Okay, that's a detection of energy transfer between two fluorophore. That's what it means. Okay, what does that mean? Okay, so let's say you make a GFP fusion. Okay, then there's another fluorescent fusion. Okay, so you're trying to see if these two proteins that are fused with maybe yellow fluorescent protein, maybe blue fluorescent protein. Okay, if these two proteins interact inside of a cell or not. Okay, the principle is if they interact, they come together and make this structure. If that structure is made, then there will be energy transfer that can be detected inside of a cell. That's what FRAT means. Transfers fluorescent energy between those two fluorophore. So you can detect that inside of a cell. I'll stop right